Well, good afternoon, everyone, and thank you for the introduction. And I want to thank the organizers and Marshall for inviting me to present here. So I'm going to show you today some of the work that we've been doing on prediction and profiling of pregnancy and child outcomes, so using machine learning and AI. So it really will focus will be on adverse outcomes and not on the cardiovascular disease. And I will start with grim statistic that there were about 2.3 million neonatal deaths, in 2023, with the highest rates in sub-Saharan Africa and South Asia. And more than half of those newborns that died were either born too small or too soon. So they were either preterm or SGA. And both of those outcomes increase risk for child mortality. Leading cause of death under age 5 is preterm birth, but also neurodevelopmental delay and growth faltering. And we looked at the growth faltering in terms of in 3 forms, in terms of stunting, which means that child has height that is too low for age, wasting, which means low weight for height, and underweight, which is low weight for age. And again, highest prevalence is low and middle income countries because of the maternal undernutrition, poverty, and infection. And finally, I just want to point out the second largest cause of maternal mortality is preeclampsia that causes about 15 to 20% of all preterm births. And why I'm giving you all these statistics at the beginning is because these are the outcomes that we have been looking at and analyzing, and more recently we are also looking at neurodevelopment and gestational diabetes. So the approach that we take in this analysis is to analyze the data that is collected early in pregnancy, so clinical demographics data and then biological samples, that then we wonder, okay, no. Just try to see there is a pointer that we use to, so we use this data to predict early these outcomes of pregnancy, and then to use also data at the delivery from the neonate. So typically we use blood spots from the heel prick and also from cord blood, and again, clinical demographics data, about the neonates, about the pregnancy, and then jointly you'll use all these data to now profile some of these outcomes and to predict the development of the child in terms of growth faltering, typically in first 1,000 days, and neurodevelopment. So this is kind of a framework that we use, and I will show you, so this is the data that we typically use in this analysis for the machine learning model. So from electronic health record, we have demographics, we have medical history, vitals, and labs, and then from biological samples, we measure various omics, whereas from dried blood spots and cord blood, we had more targeted markers that you can see here. So what we have, right, is very heterogeneous data with large number of samples, a large number of features that then we use to train machine learning models and to answer the question, well, what is the estimated risk for some of these outcomes, and what are the biomarkers that drive that risk, that determine the risk, that are most different in say, preeclampsia or not, and can we get insights about the biology and determine pathways. So this is kind of a framework, and I will show you 2 topics in the talk today, two projects that we analyzed using these approaches. One is early prediction of preeclampsia, and the other one is prediction and profiling of growth. So starting with preeclampsia, this is goal of this study, and we started this study now several years back, is to determine biomarkers that can predict preeclampsia early, before 16 weeks, before there is symptoms of preeclampsia, before there is diagnosis. And if we can do that, right, then this would enable, this would inform early treatment, right, which women should be receiving low-dose aspirin, and there is a new now promising therapy that's being developed here at Stanford. And also it will inform which women should be closely monitored, and so that they can receive timely triage if needed. And this is especially important in low resource settings, right, or LMICs, where antenatal care might be scarce and not every woman is followed closely. So we started this, again, several years back. This study was sub-study of the larger prospective study here at Stanford that was led by Prematurity Research Center and Professor David Stevenson and Professor Gary Shaw. And we focused on prediction of preeclampsia, and Neil Magapur was the senior author in this study. So in the study, it was a prospective cohort of women, pregnant women here at Stanford that were followed, and there was longitudinal data that was collected. What we analyzed here was just the sample taken early on between 10 to 15 weeks, because we wanted to do early prediction. So on those samples, you can see all the list of various omics that were measured at the time. So what this gave us was a large number of measurements of these different omics. We had about 50,000 measurements per sample at the time. So, and we wanted to analyze them agnostically, right? We did not have hypotheses. We wanted to see, well, what are the biomarkers that we can use for early prediction? So for that reason, at the time we used machine learning, and basically our approach was that for each omic separately, we would train a separate machine learning model. So for example, for proteome, I have these little orange dots, each presenting one protein, to just emphasize that there were a large number of them that we used all agnostically, without hypothesis, to train machine learning model that would then give us what we call the preeclampsia signature, that is, what are those proteins that are most predictive and how well we can predict. And then we integrated that across all the different omics. So we had various results in this study, but what is most important for my talk today is two things. Well, we saw high prediction accuracy. Again, this is before diagnosis of preeclampsia. And you can see here in this bar plot on the left, the performance. So it's in the terms of area under receiver operator curve. So if AUC is 1, it's a perfect prediction for every pregnancy. We had, at the time, AUC of 88% if we use proteome. And the other important point is that this accuracy was achieved with a very small number of biomarkers that are shown here, proteins. Some of them already known previously. You can see the first one being leptin, and you can see here in these box plots how different it is already in the first semester, and then we had some proteins that were new that we identified, and we looked at biological possibility. So that was our study. Now, based on that study, what we are doing now is that we are working on developing low-cost prediction tests. So can we, based on this small number of proteins that we measured, and we also have metabolites, developed an early test? And the first challenge that we meet is that, well, these proteins were measured using high throughput, high cost technology that measured large number of proteins, where our goal, what we want to do, is develop a simple low-cost test that can be available routinely to every pregnant woman. So obviously this kind of high-cost technology does not is not appropriate here, but it's also not needed because we know exactly what proteins to use. So our solution here in this test is to use instead ELISA technology to measure those proteins that we identified. So ELISA, it's a targeted technology, right? If you look at this one ELISA plate, it will measure one protein only. It has each of these wells is coded with a specific antigen for one protein, right? And then, and we can use it because we're only measuring few proteins. It is low cost, which is what we want to, and we want to test that is low cost, and it's also commonly used globally in the labs. So it looks like it's a good match for a low-cost test. One first question is, well, we measure those proteins with highly sensitive technology. If we now use ELISAs to measure these same proteins, can we achieve the same accuracy? So to evaluate that, we measured now these proteins that I showed you a few slides ago using ELISAs in the same cohort to see if we can now still predict preeclampsia using those proteins with accuracy that I earlier showed you. And you can see here the results. And what we've actually seen that ELISA was very sensitive in our data. It had high discriminative ability. And what we've seen that we can actually achieve that if we drop some of these biomarkers and use only 2 proteins. So what these box plots show is what is the accuracy of prediction if we use 2 proteins to predict preeclampsia. And you can see how different the prediction is for preeclamptics versus non-hypotensives. So this was very encouraging. What this tells us, right, is that that ELISA is appropriate possible solution for this test. Now, where we are right now, of course, the very important next question is we can validate now these proteins in different cohorts. So we are working right now on that. We have 2 cohorts, one from University of Pittsburgh. This is led by Joel Sadowski, who is here at Stanford working with us. And then the other cohort is a prospective cohort of 1,000 pregnant women in Bangladesh. This study is led by Child Health Research Foundation, CHRF in Bangladesh. On our side, it's led by Professor Gary Darmstadt. So where we are with this currently, in the first cohort, we have validated the first protein. We see the same difference, the same direction of intraeclamptics versus non-hypotensives. And now we are measuring the second one. There are some issues about using ELISAs in different cohorts that we are facing, but we did validate the first protein. And in the second cohort, the collection of samples and outcomes has been finalized for all 1,000 women, and now we have to start the ELISA measurements. So, and there is one more element to this test, which is based on our previous study where we developed machine learning model from electronic health records, where we observed that we can improve the accuracy of the test that I just showed you that uses proteins. We can improve it by integrating specific clinical and demographics data. So we are doing that as well. That improves the accuracy. We are making this optional because sometimes this data is not available, especially in LMICs. So what we really hope, we aim for, is that this test can be made widely available, right, for every pregnant woman during prenatal care early on, right? Because if that is so, that can drastically improve prenatal care. It can enable early intervention and improve clinical outcomes for mom, but also for the baby. So this is where we are with this study. And for the rest of the time, I want to show you our results on prediction and profiling of for gestational age, and I have also a little results to show you about LGA, large for gestational age. So this is a study done in 5 cohorts in low and middle income countries. It is led by Professor Gary Darmstadt, Victoria Ward, and David Stevenson, where data was collected in Kenya, Zimbabwe, Zambia, and 2 cohorts in Bangladesh. We have about 3,600 samples from neonates, and we have pairs, mom and neonates, so we have also about 1,100 samples from moms. And the study, if you look at the overview, is very similar to what I showed in the beginning. It's a prospective cohort. Samples were collected mid-pregnancy from moms and then from heel prick from neonates at the delivery. And we also had some clinical and demographics data. And then maternal samples were analyzed using untargeted metabolome and targeted lipidome. And heel prick, the blood samples from neonates were analyzed using newborn metabolic screening. And then those babies were followed for 3 years, actually, to follow their growth and neurodevelopment. So we have all these outcomes, as you can see on the right, and I'll just show you what we have so far for gestational age. So in terms of data, we had about 640 different lipids and the same amount of maternal metabolites that were annotated, and about 90 neonatal metabolites. You can see their groups, and you can see the lipid classes that were measured. And we applied methods from the traditional statistics. I'll show you some volcano plots, and then from machine learning and AI as well. And to discover, so what we wanted to see is are there any biomarkers, what are the profiles of SGA on these different outcomes. So we used ElasticNet, which is classical method for these kind of questions. We also used a stable, which is the method that we developed. This was in collaboration with the Briscoe Dier's lab. This was published in Nature Biotech in 2024. We compared it with neural network approaches to see if there is any gain to that, and then we trained across different cohorts using what is called invariant causal prediction. So when we did that, we wanted to, again, to see whether SGA neonates have different profile in terms of, if you look at SGA versus all the other neonates. And what we've seen is the answer is yes. So SGA neonates have a very distinct metabolic signature, meaning that the metabolites that we measured in the neonates in the SGA groups were very different, and I'll show you what I mean by that, from the rest of the neonates. And what I mean by distinct, it means that our machine learning model that we use to train had very high accuracy. So what I show, and that's shown in this plot here, on the y-axis, you have, again, area under receiver operator curve. And these different bar plots show the performance. So yellow one is if we train on one cohort, and then gray if we train on two, and blue if we train on 4 cohorts. This is a true validation. There is no cross-validation. So what we do, we train on one cohort, and then we evaluate on a different cohort. And so, for example, for Zimbabwe, we trained on Kenya data, we evaluated in Zimbabwe, then we trained on Kenya and Bangladesh, we evaluated in Zimbabwe, then we trained on all 4. And you can see that the performance, you see these bars are getting higher, they're getting, the performance gets better as we train on more cohorts, which is expected because we have more data, so machine learning model can learn more. And also we have more generalizable data, from different populations, from different cohorts. So what are those signatures? Here are the most significant biomarkers that were different, distinct between SGA and AGA. So for example, you can see that alanine was, so I'm showing it here as a function of gestational age of the delivery, and you can see that in SGA group, alanine was higher consistently compared to the other neonates. And then, for example, if you look at the bottom, we have the ratio of C3, C2. This is propionylcarnitine and acetylcarnitine ratio. And you can see that it changes with the gestational age, the delivery somewhat, but it's always lower in SGA group. So this now you can see how this can now give you some insights or help you maybe create hypotheses about the biology and what happens. So for example, a hypothesis why alanine would be higher is because it is used in glucogenesis and this pathway is not functioning properly in SGA. So alanine is not being used as it would be otherwise. So you can see really levels of alanine that accumulate and so forth. So what really this tells us, this type of results, what really help us is to characterize SGA beyond the size of the neonates, right? It can tell us, it can really give us insights into metabolic changes. So we did this same analysis in a very different data. This was led by Daniel Dyatlov. He was doing a project in my lab. He's a medical student here at Stanford, where he analyzed about 41,000 singleton live births, okay, in California biobank data. So, so the question was the same: can we see the SGA profiles? But the cohort was very different, right, and very different population. And what is the strength of this data is that because it's so large, we could look at all these different subgroups. For example, we could look at the preterm SGA, the most vulnerable newborns, which we did not have in the LMIC data. So what we've seen when we looked at the neonatal signature is it was very consistent with what I just showed you in the LMIC's data. But here, again, we have these different groups, and one thing that we can observe, like from this table, that neonates that are more affected had more distinct metabolic signature. You can see that the preterm SGA neonates have higher AUC, meaning their profiles are more distinct from the AGA group compared to the, say, SGA 10%. And on the right, you can see again what are the metabolites that were most distinctively changed. These are on the y-axis, and this on the x-axis, I'm showing the coefficient from machine learning algorithm, and larger the coefficient in the absolute value, it is more different between SGA and AGA. And you can see that most predictive or most change was, again, the ratio C3 over C2 that I showed a few slides back in the other cohort. And also for the other, there is another example for term SGA versus preterm SGA. So again, you can have the hypothesis why this could happen that would offer some insights into biology. So some of the reasons here is that impaired fatty acid oxidation can lead to accumulation of the acetylcarnitine, therefore reducing the ratio. and that the specific nutritional deficits can affect the propionkalnitin levels, again, reducing the ratio, and we have a few more analyses in that, along these lines in the paper. So when we did this analysis, what we've noticed, because we integrated it with the clinical data, is that preeclampsia really alters the profiles that I just showed you. So in other words, if the profiles that I just showed you will depend a little bit whether the mom had preeclampsia or not. So we looked at that more closely, and we observed that neonates that are exposed to preeclampsia have very distinct metabolic profile. We did not look at the SGA in this analysis, solely on the preeclampsia, and we saw distinct metabolic profiles. We were not first to observe this. This was already shown in the paper by John Rees and David Stevenson, Gary Shaw, and Neem Agapur, where they observed the same thing. We then looked a little bit further, extended it, and to different stratifications. And what we've seen is that you can see that for superimposed and severe preeclampsia, these AUCs highest, this is where we saw really distinct profiles. And so this suggests that, perturbations that happen in pregnancy in mom really leave detectable signature in the newborn metabolome. That is what this result really tells you. So, and we looked at it, we stratified this model by neonatal sex to see if there are differences. And we actually did not see much difference either in performance. You can see that if we look at by females and by male, the AUC is almost the same. It's within the confidence intervals. And if you look at the coefficients for two, female in red and male in blue, they are very similar with the two main metabolites being the same section. So it was very robust across stratification, this model. So, and the last thing that I want to point out is that we tried this model in two different cohorts, in Canadian cohort and Bangladesh cohort, and it validated. We could still see it really applied, from our California data to these two different cohorts. So this was encouraging. So again, we think this provides better characterization of SGA beyond size. And it suggests that there are some fundamental metabolic consequences of fetal growth compromise in those neonates. So, almost done. I just want a few words to say that we are now working on profiling of LGA using the same methods and the same data. This is led by Ananya Sirivasiredu. She's a student in my lab. She's a master's student in computer science. And she developed a model now for LGA. And again, we see the ratio C3, C2 as the best predictor, but now we see it increased in LGA. And we are now extending these to look at, in those now children, the consequences of LGA in terms of obesity, diabetes, and cardiovascular disease. And just to point out that so far, I showed you profiling of SGA, right? We used neonatal samples at birth, when the baby is already born. But can we actually identify any changes that precede SGA? And we looked at that, and I will just end with that. In the same study from the LMICs, as you remember, we also measured we had maternal samples. So we looked whether these samples can tell us something about the small for gestational age and for preterm birth. And this is the result we had for a maternal lipidome, how it looks in preterm birth and preterm birth pregnancies. And you can see that this volcano plot is highly skewed, right? You see that what you see is a really strong lipid imbalance where lipid levels are increased in preterm birth. And if we look at what are the classes that were increased, they were mostly triglycerides. So I'm plotting the same plot, but in terms of lipid classes, and you can see that most of them are yellow, meaning that these are triglycerides. And we looked at then, so what I'm looking here is each association of each lipid at preterm birth. When we looked at them by class of lipids, that is shown on the right, you can see we looked at saturated, polyunsaturated, and monounsaturated. And for each class, the odds ratios are consistently higher in preterm birth. So really confirming what you see on the left. And then in SGA, they were really opposite situation where these lipid levels were lower, and again changes were in triglycerides, but signal was much lower, not as strong. You can see that there is less of these red dots, the ones that are statistically significant. So, but again, all the ratios were all lower than 1, as you can see to the right. So and I'm not showing you machine learning algorithm results because they were not very predictive. The AUC was like 0.64, so they are not, not be used for prediction, but definitely there is a signal that precedes SGA already in pregnancy. That's what these results tell you. So just in summary, we see that omics in clinical data in pregnancy and at birth can predict adverse outcomes. We are very hopeful about our low-cost ELISA test. And we are now have some results and a lot of planned future work to extend this analysis to a 2-year growth in terms of stunting, wasting, and underweight, and also neurodevelopmental delay. And just to thank my wonderful collaborators, starting with Professor Gary Darmstadt, Victoria Ward, and David Stevenson, and our colleagues here at Stanford and at the global sites. And thank you all.